Hello! Today we have an interesting practical work. We will solve the problems of molecular biology. Tasks with the structure of proteins and nucleic acids. Click on the model of the structure of the formation of the macromolecule. Remember how proteins are formed. Let me remind you, proteins are polypeptides, the monomers of which are amino acids. The relative molecular weight of the amino acid is 100. The length of the amino acid is 0.35 nanometers. Click on the picture of the nucleic acid. Look at what remains it is built from. Nucleic acids are polynucleotides consisting of nucleotides. DNA. It consists of two spirally right-hand twisted chains. Click on the DNA molecule. Each nucleotide contains a deoxyribose pentose, an orthophosphate acid residue, and one of the four nitrogenous bases, A, T, G, and C. Remember these letters. Click on them. Reckon the numbers of bonds between the nucleotides. Look at the lines. 1 dash equals 1 of linkage. The distance between neighboring nucleotides in DNA is 0.34 nanometers. There are 10 pairs of nucleotides per one turn of the spiral. So, the pitch of the spiral is 3.4 nanometers. Relative molecular mass of nucleotide is 345. And now, let's study the main rule of molecular biology, Chargaff rule. The amount of adenine is equal to the amount of thymine, and the amount of guanine is equal to the amount of cytosine. A equals T and G equals C. Adenine equals thymine and guanine equals cytosine. Hence, A plus G equals T plus C. That is, A plus T plus G plus C is 100%. Adenine plus thymine plus guanine plus cytosine equals 100%. Click on the model of RNA. RNA. Let's remember that it is a single chain molecule. Instead of deoxyribose, it contains ribose and does not contain thymine. Uracil takes its place. Now, let's move on to the most interesting part, namely the tasks. Problem 1. The molecular mass of pepsin is 35,500. Calculate the length of the primary structure of this protein. The solution? 1. Determine the number of amino acids in a polypeptide, remembering that the molecular weight of one amino acid is 100. The number of amino acids is indicated by the Latin letter N. N is 35,500 divided by 100 equals 355. 2. We find the chain length, knowing that the length of one amino acid is 0.35 nanometers. 355 times 0.35 is 124.25 nanometers. The answer, the length of the primary protein structure, is equal to 124.25 nanometers. Click to continue. Task 2. The fragment of the first strand of DNA has the following nucleotide composition: GGG, CAT, AAC, GCT. Determine the sequence of nucleotides in the corresponding fragment of the second chain. Here, let's recall the structure of DNA and Chargaff's rule. Let's move on to the solution. 1. Write down the sequence of nucleotides on the second strand of DNA according to the complementarity rule. DNA, GGG, CAT, AAC, GCT, the first chain. We're completing the second one. CCC, GTA, TTG, CGA. This is the second chain. You see, everything is quick and easy. Simply arrange the letters according to the rules.